record today's lecture as well, just in case I go through the slides quickly. And also there's students who are writing the quiz off-site, and they may not get here in time, so I figure might as well do that. The only problem is I see full screen here. On my screen, I see presenter view. I'm trying to figure out a way to just use slideshow. There we go. Got it. Perfect.
From last class.
And I'm going to try to get rid of
the
this.
slides.
Hide floating meetings control. Good. There we go. That way,
Wait.
what I see on the screen is exactly what you see on the screen. Perfect. So last week I finished talking about AP 1209, which is an antisense oligonucleotide, the TGF-beta ligand. So this is where I ended off last week. And I explained how, where I went through the experiment where if in untreated cells, you seek them down, these are pancreatic tumor cells, they'll actually just spread to grow into a colony shown over here after 65 hours. If you add exogenous TGF-beta to it, it'll grow faster. If you go in with an antibody to TGF-beta, you cannot block the growth. It looks exactly like the control, and that's because the theory is that the tumor produces it, it's secreted, and it binds to the receptors and signals before the antibody can even see the TGF-beta and block it. So what the authors went in with AP 1209, which is an antisense oligonucleotide. It identifies the mRNA for TGF-beta, causes it to be degraded. No TGF-beta mRNA, no TGF-beta ligand it's secreted, and therefore it cannot be bound to the receptor, and it cannot signal. And the tumor cells will stop growing. So that's the idea. In vitro, they actually took it to phase 1/2 clinical studies, and this was kind of interesting because normally in phase 1 clinical studies, what you do is you want to make sure that the drug's not hurting the patient. That's the aim of that clinical study. In this case, you'll actually start to see positive results, which are very, very promising. So basically, if you look at, let me get a laser pointer. If you look at the patients that they had in their study, there's two studies here. One is for anaplastic astrocytoma. The other one is for glioblastoma multiforme. So AA versus GBM, shown over here. This is the patient number. This is the age of the patients, the sex of the patients, and they also looked at the Karnofsky performance status. KPS is basically how sick is the patient. It's a standardized or sort of. Yeah, it's a standardized test. 100% means perfectly healthy, fine. We're at 100% in this room because we're able to come to the lecture. I can talk, you can listen. So it's perfectly fine. If you go 0 is death, and anywhere in between is how well you can carry out your daily functions. So 100% is great, 90 is still fine. When you start to get down to the 70s, that's when you start to get a little more ill. You're impeded from doing everyday sort of activities. And then this is the tumor volume. The previous therapies, which is either surgery or tamoxifen plus Calyx, all these other sort of chemotherapeutic agents. And they decided to also go for a number of AP 1209 cycles. And this is the survival after recurrence. Now, I forget which one's which. Let's see. Oh yeah, okay. So, anaplastic astrocytoma, the survival rate is 42 weeks on average, about 6 months. And if you look at the AA patients here, some were 32 weeks, unfortunately, but others were up to 146 weeks. And GBM, it's lower than that. It's about 23 weeks, 24 weeks. So it's a few months, maybe 4 months, 4.5 months. And some of them actually went up to like 276 months. So there are, first of all, the drug was well accepted by the patients without any side effects. And some patients actually responded quite well to it. So here's another example of the anaplastic astrocytoma, grade 3, 42 weeks, median survival. It goes up to 146 weeks if the if these patients are treated with AP1209. So tripled their lifespan, which is fantastic, and doubled the lifespan of glioblastoma patients from 23 to 32 weeks to 44 weeks on average. That's the mean survival after. And what's interesting is some patients, like this patient in blue here, this is patient 1-04, only got one dose of AP1209. Now the way they have to do it is actually have to make a burr hole into the brain, or actually into the skull, to access the brain, and then they dripped in this antisense oligonucleotide into the brain, right into the tumor of these patients. And so they did it once for this patient. This patient got multiple multiple cycles of AP1209. They did 2, then they waited a while, and then did a whole bunch more. And I'll show you why in the next slide. And that's patient 117. So here we have the 2 patients. So we have surgical resection and radiation. So you have this empty spot here. That's where the tumor was. That was removed. And unfortunately, you see these are other lesions, i.e. other tumors. They put a catheter, was placed right here, right next to this tumor. And they dripped in the 1209. And then after 4 weeks, 1209, the tumor actually grows a little bit. After 10 weeks, it grows even more. And then after 6 months, it starts to resorb and get smaller, and then the tumor is gone after 17 months of 1209. So this is one of the patients. I think this is patient 1-4 up here. This one's 117. So it also happens whenever you try to go into the brain like this. Here's the resection. Of course, they didn't get the full margins, right? You got to get the margins, not only the tumor, but everything around it. They didn't get the full margins, and so they left, unfortunately, a little bit of tumor down here. And that's when they started the catheter placement with 1209. So here's a tumor, and this dotted line down here with this sort of discolored area, that's an edema. So that's edema within the brain. Very dangerous. So what you have to do is you have to stop anything that you're doing. So they stopped the 1209 treatments, and they waited a while, and then they restarted with the 1209 treatments after the edema had resorbed into the cerebrospinal fluid. And it goes up to like F over here, which is basically this patient right here. So then they continued a whole bunch of treatments. So the tumor actually got larger. So here's you have the edema. Tumor continued to get larger. They continued with the treatment, and then the tumor got smaller and smaller, and as you can see it over here. So it is quite effective in a small number of patients. Question?
Why does the tumor get bigger first?
Oh, because, well, what happens is the tumors continue to grow, and what you want to do is you want to block the TGF-beta production, and as you're doing that, the tumor sort of slowing down, and then it'll start to resorb. So it'll continue to grow, and that's true of any chemotherapeutic agent that you use. Tumors tend to grow larger and larger and larger, and then hopefully the drugs kick in and start to kill the tumor cells, and they get smaller. Yeah. It also happens with that tumor flare that I mentioned at one of my first lectures. I talked about tumor flare when you go in for people with prostate cancer, for example, where you go in with GnRH. They're super agonists. What they do is they bind to the receptor, and because of that, they actually signal improperly, and then the tumor gets larger and larger, and then eventually you downregulate the receptors, and then you get rid of that sort of thing. So a lot of chemotherapeutic agents don't act immediately. The tumor gets larger and then gets smaller. So that's 1209. I talked about the antibody, which is okay but not great. 1209 is very promising, but what about kinase inhibitors? Can you actually block the TGF-beta signaling? Just go in with kinase inhibitors. They're small. They can actually get into cells much better than antibody. Well, antibody can't get into cells. And in 1209, you actually have to make burr holes, for example, into the brain, put in a catheter to get that across or get into the cells. Can you make an inhibitor that can either cross the blood-brain barrier and or get into as many cells as possible? And here's an example, SB431542. This is the canonical TGF-beta receptor inhibitor that was published about 20 years ago, almost 20 years ago now. And it is an excellent inhibitor. We use it all the time in the lab, in cells for sure. It sort of looks like ATP. It's a TGF-beta type 1 receptor inhibitor. So I might as well start drawing this up on the board. We'll have the type 2 receptor, type 1 receptor. Here's TGF-beta. So this is the type 1 receptor right here. And so the inhibitor actually goes into the ATP binding pocket. And that's what blocks. There we go. It goes into the ATP binding pocket right there. So it's a kinase inhibitor. Now it's a serine-threonine kinase inhibitor, not a tyrosine kinase inhibitor, because the receptor is a serine-threonine kinase. And the IC50 is 94 nanomolar, which is quite good. And I showed this sort of table last class, I believe, and it goes through how you would actually use it, or what were the steps that you would use to actually see if the drug is actually working. So I'm just going to go through a lot of the different steps that are on this table and show you, using the primary data from the paper, that actually characterize this inhibitor. So the first thing that they did was they looked at, and here's the take-home message. Let's see if I can minimize this or move it down here so you can actually see what the titles are. So SB431542 blocks TGF-beta-induced complex formation between SMAD2 and 3, SMAD4, and nuclear translocation of SMAD2 and 3. So that's the take-home message from this slide. How did they come about or come to this conclusion? Well, they started out with a number of different cells. So they had 293 T cells. These are human embryonic kidney epithelial cells. They had A549 cells. These are human lung adenocarcinoma cells. And FETs, human colon cancer-derived non-tumorigenic epithelial cells. So they use 3 cell lines here. And what you do in these 3 cell lines is they all have TGF-beta receptors. So you serum starve the cells. So where you see net minus minus here, minus 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 minus, that means that these cells were serum starved. There's no TGF-beta in the serum because you remove the serum. There's no TGF-beta in the media. And if you look for the phosphorylation or the signaling, what you have to do is you have to add in exogenous TGF-beta. So they added the TGF-beta here. And the second you add TGF-beta, you get phosphorylation of SMAD2. So SMAD2, because the receptors phosphorylate SMAD2, and I'll just draw a phosphate there. So we don't have antibodies that look for and can identify activated receptors, so we do the next best thing. We go and see if the substrate is phosphorylated or not. If this is not phosphorylated, that means that this cannot be active. So you add TGF-beta, you get nice phosphorylation of SMAD2 in all 3 cell lines, over here, over here, and over here. And then you can start titrating in 2 or 10 micromolar of this SB compound. And you can see that the phosphorylation of SMAD2 is blocked in the 293 T cells, it's reduced in the FET cells, and it's blocked at the highest concentration in the A549 cells. So using this, they looked at the phosphorylation of SMAD2 and they assessed using phosphospecific antibodies. And indeed, if you go in with into 3 different cell lines, 10 micromolar SB compound is very potent at inhibiting TGF-beta receptor activity. Next, if SMAD2 is phosphorylated, at that point it'll associate with its common SMAD, its partner, which will be SMAD4. So what you can do is you could isolate SMAD2 and see does SMAD4 associate with it. And the way you do it is you go in with an antibody to SMAD2, for example. So here's SMAD2, and let's say TGF-beta signaling is occurring. So you add a TGF-beta, and SMAD2 is associated with SMAD4. So what you can do is you can go in with an antibody, like a SMAD2 antibody, attach it to beads, which will precipitate, and remove the SMAD2 from the lysate, from all the proteins in the lysis, when you break open all the cells. And then you wash off the beads, and then anything that's associated with SMAD2 will also come down. So if there's no signal occurring, SMAD4 should not be associated with SMAD2, so you only see SMAD2. But if you add TGF-beta and you allow this to occur, not only do you bring down SMAD2, but you also bring down SMAD4. So SMAD4 comes along with the beads and is isolated. And you can purify both of them. So here, what they did was they immunoprecipitated SMAD2, and they looked for SMAD4. And if you have no signal, shown over here, and over here, serum star cells, there is no phosphorylation of SMAD2, there's no association with SMAD4. But then you add TGF-beta, and if you precipitate SMAD2, SMAD4 also comes down. And that's why you have a band for SMAD4, if you Western blot for SMAD4. So there it is, in the 293 T cells, FET cells, and the 8 factor 9 cells. And then if you go in with the inhibitor, you block this association, because you block the phosphorylation of SMAD2, no more association. And of course they had loading controls here, which is really nice. They had SMAD2, they threw in SMAD3 here, which is nice. They didn't have to, but they did, just to show the relative levels of this protein and the relative levels of SMAD4, which is also nice, and beta-actin is another loading control. Now, if this complex occurs, it's going to go into the nucleus. So that's the next step that they looked at. Can this complex go into the nucleus in response to TGF-beta? Can you block it with the SB compound? And the way they do it is they did subcellular fractionation. So C stands for cytoplasmic, and N stands for nuclear fractions. And they look for accumulation of SMAD2, SMAD3, and SMAD4 in the nucleus versus the cytoplasm. And here we see that, let's look at SMAD2. SMAD2 is a good example. So 0 hours means no incubation of TGF-beta. So the majority of the proteins should be in the cytoplasm. There should be nothing in the nucleus. You add TGF-beta for 30 minutes, and you have nuclear accumulation of SMAD2 and SMAD4. It's higher than the control. And then you go in with increasing concentration, or not increasing concentration, 10 micromolar of SB compound, and you block the nuclear accumulation of SMAD2, you reduce the nuclear accumulation of SMAD3, you reduce the accumulation of SMAD4. And that's true at half an hour or 1.5 hours of TGF-beta induction. And you can prove that these are cytoplasmic versus nuclear fractions because they used a cytoplasmic protein to show that the cytoplasmic fractions are clean, and the nuclear protein to show that the nuclear fractions are clean. So good use of specific compartments, markers. So using several different techniques, immunoblotting using antiphosphatyrosine antibodies, columnar precipitation of SMAD2 with SMAD4, and subcellular fractionation to assess nuclear accumulation of SMAD proteins, they come up to this conclusion. Now, if you look over here, it looks like SMAD3 and SMAD4 already start out in the nucleus. So these results, although quite good, shows you a limitation of doing the experiment by subcellular fractionation. This is true not only in this paper, but even in our lab when we try to isolate nuclear versus cytoplasmic fraction. Sometimes it's a little messy. So they decided to do it using a second technique, immunofluorescence microscopy. So here, they looked at nuclear translocation of SMAD2 using immunofluorescence microscopy. And this is quite interesting. If you start off with a cell that would be the nucleus. What happens is if you do, and they use red, so I might as well go with red. They went through the SMAD2 antibody. In a quiescent cell that has no TGF-beta, because SMAD2 is everywhere in the nucleus, it's sort of hazy. It's hard to see, and it's sort of hazy. And that's what you see up here in the control. Hazy cells. And the cells themselves are kind of like fried eggs. So if you put them sideways, they'll actually look like this. So this darker haziness that you see over here, over here, over here, that's basically a lot of sort of hazy SMAD2 when you're looking down that way. Now the second you add TGF-beta, all of this TGF-beta concentrated into the nucleus, you get a nice bright nuclear spot. Why? Because you phosphorylate all the SMAD2 that's floating around in the cytoplasm and it accumulates into the nucleus. And that's what you see. Minus beta plus beta. And that's what they see here. So here you have the hazy, everywhere sort of SMAD2 signal. You add TGF-beta and it accumulates in the nucleus. And you can tell it's the nucleus because they use DAPI to show you where the nuclei are. Then you go into the inhibitor, the SP compound, back to haziness. Why? Because you block the phosphorylation of SMAD2, cannot associate with SMAD4, doesn't go into the nucleus. And that's true of both SMAD2 and SMAD3. They wanted to look at both of them. So here, this one figure, this is still figure 1 in this paper. In this one figure, they used 4 different techniques, 3 different cell lines to assess the ability of this inhibitor to inhibit TGF-beta receptor signal. Just the signal alone. So, once you get a signal going, SMAD2 and SMAD4 are transcription factors. Because they'll bind to transcription factors. SMAD2, SMAD4. And you can actually look at this, you can look at it endogenously, or you can go in with reporter assays, reporter constructs, which are basically plasmids. So, let me move this over. So you have FET, these are human colon-derived non-tumorigenic epithelial cells, or HepG2 cells. These are liver carcinoma cells. And what you do is you can transfect in plasmids that has luciferase. I think I explained that last week. So basically you'll have your luciferase DNA, cDNA, will actually produce luciferase. And then you hook up to that a promoter. And that promoter can have, well, there's different promoters you can use. They used 3TP, which is 3-TPA pi1 promoter. CAGA, which I discussed last week, CAGA, pi1, or P21. All of these proteins, either endogenous proteins like P21 and pi1, or these artificially constructed CAGA repeats, or the 3TP, all of them respond to TGF-beta. So these are all promoters that respond to TGF-beta. And so here we see that in the absence of TGF-beta, there is no luciferase activity. No light is given off if you add the substrate because there's no signal. You add TGF-beta, you get a robust signal. And over here, the 3TP lux, the CAGA lux, P21 or pi1. And then if you titrate in the inhibitor, you reduce the luciferase here, 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 and here. So using 4 different TGF-beta sensitive gene promoter reporters and 2 different cell lines, you have to show that SP inhibitor inhibits TGF-beta transcriptional activity in a dose-dependent manner. So this is an artificial transcriptional activity assay, something you do right away to see if it works or not, if your inhibitor works. And if that works, then you can go and do more complicated screens and assays, which is to actually look at the protein. So now that we've proven that the SP compound, SP blocks the type 1 receptor because it blocks the phosphorylation of SMAD2. It blocks the association of SMAD2 and SMAD4. It blocks the nuclear accumulation of the complex. And it blocks the ability of SMAD2 and SMAD4 to generate, well, transcribed DNA, causes transcription of DNA. The next step is to look for protein, because that's next downstream of protein. That's downstream of transcription. And which proteins you want to look at? Well, you want to look at TGF-beta specific proteins. So pi1, fibronectin, did I write that down? This is fibronectin, this is pi1. Should probably I didn't write that. 3 different proteins that are usually expressed. Yeah, I don't have it. Oh, there it is, fibronectin. And Pi1 is plasminogen activator inhibitor. Perfect. So I did write it down. So here's Pi1, fibronectin, and of course the loading control is beta actin. In the absence of TGF-beta, there is very little Pi1 expressed by cells. There's a little bit of fibronectin, and a little bit of Pi1, and even P21. There's a little bit there. You add TGF-beta, all of these expression levels go up. These are proteins. And then you can go in with your inhibitor, SP compound, to block the expression of Pi1, reduce fibronectin, reduce Pi1 here again in different cell lines. This is red intestinal epithelial cells and P21. So using 2 different cell lines, they show that you can block the protein expression using this SP compound. So once again now, you block protein expression. Now, those are some of the proteins. Yeah, question?
Yeah, sorry. The previous slide, in panel A, why did they move from 2 to 10 micromolars of SB and then the cyclinated 20?
That's a typo.
Oh.
Good catch. So this is 2, and this one should actually be here. That's a typo. Yeah. Yeah, that's a good catch. Thank you for pointing that out, because that would have been, students would be writing, what's going on here? Yeah, that's what happened. So this paper, like I said, went through all the authors, people who read it, the reviewers, the copyright editor or the copy editor, and everybody missed it, and it was published like this. Yeah, happens all the time. More than you would expect. So if you find little typos in papers, don't be surprised. I have my own papers that also have some typos, which I'm not proud of, but they're not detrimental, they're not deadly, they're not, oh no, the whole thing falls apart. You don't have to retract anything, it's just like, yeah, it happens. Yeah. So, they showed that you can block protein expression, but TGF-beta induces a specific outcome in epithelial tumor cells, and that is EMT. So instead of looking at just general protein expression, let's see the proteins that are expressed in response to TGF-beta in the EMT. So here, they look for EMT proteins. Well, first of all, they said, okay, do the cells look epithelial versus mesenchymal? So they did bright field microscopy. Here are control cells. These are NMUMG cells or PANC1 cells. So they grow like little islands because they try to make little epithelia. Even though they're tumor cells, they try to make little epithelia. So they grow as little islands. And they like to stick to each other because they're epithelial cells. But you add TGF-beta, and they start to sort of fall apart. They don't want to touch each other anymore. They go rogue. They go, their own way. Shown over here and over here for these 2 different cell lines. And then if you go in with the SP inhibitor, you come back to epithelia again. So these cells grow in islands. And you can do that just by looking at the bright field, but you can also look for proteins that are found in the belts that hold the cells together. Adherence junctions or the tight junctions. And you can see right here by immunofluorescence microscopy, you make little belts around cells. This is ZO1 or eucadherin. This is a tight junction protein, ZO1, or eucadherin is an adherence junction protein. Both of them make nice little belts. And then you add TGF-beta, and so those belts sort of fall apart because you're getting rid of these tight junctions and adherence junctions. You go in with the SP compound, and you have the nice junctions again, the belts around the cells. That's true in the NMUMG cells. It's also true in the PANC1 cells. Shown over here. And you don't have to only do it by immunofluorescence microscopy. You can also do it by Western blotting, shown down here. So here in the absence of TGF-beta over SP, you have lots of eucadherin produced, lots of ZO1 produced. You add TGF-beta and you lose your ZO1, and then you increase the concentration of the SP compound and the ZO1 comes back. Now, notice, eucadherin gives the inconclusive results. And that is because they incubated these cells, I believe, for only 24 hours with TGF-beta. And you need more than 24 hours in order to sort of block the production of eucadherin. so that you can actually cause the breakage of the epithelial cells, sort of the adherence junctions. So what they did was they went in and used a longer time course with these cells. So instead of going in with only 24 hours of TGF-beta, so here's the amount of ECAD adherent you see in the absence of any stimulation. You add TGF-beta for 24 hours, you still have lots of ECAD adherent. But then after 48 hours and after 60 hours, now you start to see the decrease that you expect during EMT. And that loss is blocked by the SP compound. So because these results here were inconclusive, they said, let's do the time course longer, shown over here. And indeed, EMT is occurring in cells with TGF-beta only, but then you add the inhibitor, and you block the loss of ECAD adherent. You didn't have to redo this with ZO1 because ZO1 already really nice reduction here with TGF-beta. And then that's also blocked with the SP compound. So in this case, they used two different techniques and two different cell lines. The author showed that SP 431542 became this TGF-beta dependent EMT. All right, so the protein here now that they've showed was E-cadherin and ZO1. These actually were epithelial markers. They got rid of those. Now, if the cells are not, if the cells have undergone EMT, they're not epithelial. They're not all attached to each other, and because of that, they're free to migrate. So if you have a mesenchymal cells are migratory, epithelial cells are not. So the question they asked next was, can you actually can you block the TGF-beta dependent migration of these tumor cells using this SP compound? And here, they used A549 cells again. These are lung tumor cells. Or they used MB231, the breast cancer cell line. And they looked for cell migration. So here we have migrated cells on the y-axis versus conditioned on the x-axis. If you have no TGF-beta, there is very little migration. There's some migration, but not much. You add TGF-beta, you induce the migration, you push the cells to undergo EMT, and now they're migratory. And you can block this with the SP compound, 2 or 10 micromole. And that's true of the A549 cells. You can also do this in the NDA231 cells. These are slightly different techniques. The top one, what you do, it's called the Transwell technique. So what you do is you put media on the top, media on the bottom, and you put your cells on the top part of this insert. So this insert is literally looks like that. So basically, it literally sits inside a well. And what you do is, because you have your insert here, you put all your cells down there on this side. This insert is actually a sponge or a filter. So there's a filter that has holes in it. And if the cells are migratory, what they'll do is they'll walk along the filter, they'll find a hole, migrate, and go to the other side. Migrate and go to the other side. And then you can count how many cells are on one side of the filter versus the other side of the filter. That's called a migration assay. Any questions on this? Do you want me to explain it again? I can explain it again. Okay, so how many cells have been able to migrate through a filter? And so what they did was they counted the number of cells on the bottom part of the filter. Here they had 20, here they had 80, or about 80. And that's blocked with the SP compound. That's one way to do a migration assay. The second way of doing a migration assay is easier. Let's say you have a dish of cells, and you wait for them to be nice and confluent, and then you literally go in with a pipette tip, and you scrape a whole bunch of cells right in the middle. You create a wound. Shown over here. This wound right here. That's why they just scrape the cells right off the plate. And then you wait for the cells on either side of that scrape to sort of migrate in. That's a wound scrape assay or wound healing assay, as it's called. And you add TGF-beta, and you see that there are more cells, shown by the bright dots here, that have migrated into that wound. And then if you go into the compound, the inhibitor, you actually block that migration. There's fewer cells in that wound that have migrated. So that's just migration. Now, as you saw in the quiz, you can migrate, but you can also have to invade by going through a basement membrane, through MMPs, and you can actually test that in vitro as well. So this Transwell assay, let's draw this again. So here's the filter with the holes in it, with the pores, they're called pores, they're 8 microns across. And if you plate the cells on top of this, you're just looking at migration. Or what you can do is you can coat this filter with Matrigel. And then place your cells on top of that. So these cells have to chew their way through the Matrigel. They have to eat their way through the Matrigel. Find the pore, and then migrate to the other side. Find the pore, and then migrate to the other side. Okay. So it's not just a quick and easy migration assay. They have to eat their way through the Matrigel in order to get to the filter, in order to migrate to the other side of that pore. And then what you do is just scrape off these cells and count how many cells have migrated, actually invaded. This is called an invasion assay. Okay. So you can either coat your filters with collagen, or you can coat it with Matrigel. In both cases, the cells have to eat their way through the collagen or through the Matrigel to get to the other side of the filter. In the absence of TGF-beta, you got very few cells invading. You add TGF-beta, it goes up in both cases, and then you go in with the SP compound and you reduce the invasion of these cells. So here, using multiple cell lines and techniques, i.e. cell invasion in panels A and B here, or cell invasion, cell migration, excuse me, panels A and D, or cell invasion, panels B and C. This makes a convincing analysis. I thought so. All right. Then the last thing they did, and this must have been asked by the reviewers because this was off of my radar. This is not the kind of thing I would normally do. They actually went in and said, okay, let's see if TGF-beta, what does TGF-beta do to anchorage-independent growth? So how a tumor would grow inside a tissue that's not attached to a hard piece of plastic. So this is as close as you can get to an in vivo assay. So they went in with A549 cells. Now what's interesting is TGF-beta normally blocks A549 cell growth. Okay. They went in with HT29 cells, and TGF-beta normally stimulates the growth of these cells. Okay. And then they went in with a third cell line that is indifferent to TGF-beta. It doesn't care if TGF-beta is there or not. It will not be affected. The growth of these cells will not be affected in anchorage-independent growth in the agar, soft agar, for example. So basically, what are you looking at here? In the control cells here, there's TGF-beta. And TGF-beta normally blocks the A549 cell growth, and so you have very few colonies. You go into the SP compound, and you get larger colonies over here. Colonies are much, much larger. Let's move to the opposite end of the spectrum here. These cells normally like TGF-beta, and they'll grow with TGF-beta, so you get these huge colonies. You go into the SP compound, and you block the growth. And then this third cell line that couldn't care less whether TGF-beta is there or not is indifferent to the drug. You go into the drug, you don't go into the drug, cells don't care because it's not TGF-beta dependent anyways. So the drug's not blocking their growth because it's doing something weird. It's specifically targeting the TGF-beta receptor. That's what the point of this graph is showing. These graphs are a little complicated. They're complex ways of showing the exact same thing that I just described up here. But down here, this is a nice confirmation of what I described up here. So basically, minus SP plus SP, the E549 cells, they're normally number of colonies. You go in with the inhibitor, you get larger colonies shown over here. This cell line that doesn't care about TGF-beta, plus and minus the SP compound, no change. This one loves TGF-beta, so if you block it, you actually block the growth. So here we have, the figure shows that SP compound inhibits TGF-beta dependent stimulation or inhibition of cell growth. The authors not only use cells whose growth is stimulated or inhibited by TGF-beta, but they also use one that's indifferent. And that's to show that the specificity of the drug's not having these weird off-target effects that is independent of TGF-beta receptors. Everything you see in the paper, including the growth here, has to go through the TGF-beta receptors. And that's what they're proving here by using this third cell line that continues to grow whether the drug is there or not. So basically, you've gone through all of this. In fact, they did a lot of phospho-SMAD analysis, gene expression, P21, PI1, proliferation. They looked at migration, EMT. So the question next is, SB4, well, let's just use a cell culture summary of conclusion so far. SB431, 542, extremely effective inhibitor of TGF-beta signaling and downstream effects. It has passed all the cell culture characterization stage with lots and lots of different techniques. So then the question is preclinical studies. They went in, they looked into animals. Is it bioavailable? Is it water soluble? And it ends up killing all the animals. So that's a problem. So you spend all this time characterizing a fantastic inhibitor, only get to the last stage and you can't use it in an animal because of the toxicity effects. So then what you do is you go to your chemists and you say, okay, this is quite toxic to animals, what can we do? And they start to put bells and whistles on the drug, on the compound, to make it less susceptible to undergo oxidation or to hit different systems. And what they do is they did minor modifications. Now, the molecule on the right here doesn't look at anything like the molecule on the left, unless you actually look at it a little closer. So, this CONH2 here, that's the CONH2 right there. This group is identical right there. If you take and you spin around this bond, so this top group here becomes the bottom group, and this bottom group becomes the top, you see that it's very, very similar. So just take this group here, flip it 180 degrees. This is the same as that, and this is the same as that. Or very similar. This is very similar to that. There's an extra CH3 there. This is identical, and then they added an extra 2 carbons, or sorry, they added an extra 2 carbons here that we're missing in the benzyl group there. But having said that, this looks very similar to that. This is well tolerated by animals. That one isn't. Just small bells and whistles to make one. And the inhibitor, the inhibitory factor, like IC50, is even better than the original SB43152. So now let's prove that it's actually better in an animal study, or it's good, it works in an animal study. Well, first what you do is you want to make sure that it works in cell lines. So you took 4 T1 cells. These are cell lines derived from spontaneous memory tumor-involved C mice. Cells are grown in culture. Absence of TGF-beta, or absence of anything. No serum, serum starved. Very low RSMAT signal. Very little phospho-SMAD2. Shown over here by phospho-SMAD2. You add TGF-beta exogenously. It gets phosphorylated, and you titrate in different concentrations of this SM16. To the side here. Very important, soon. SM16. And you actually block the phosphorylation of SMAD2, indicating that it is working. And they did a whole bunch of other studies. I figured I might as well just jump to the punchline, which is great. What they did is they wanted to look at 4T1 tumors in animals. So what you do is you take the 4T1 tumor cells, inject them into the mammary fat pads of animals, of mice. You wait till the tumors are 9 to 10 square millimeters in size. Sideways in area. so they're palpable, you can actually feel them. Then they went an intraperitoneal injection, 10 or 40 milligrams per kilogram body weight. So this is the tumor. And you get the primary tumor, which is in the mammary fat pad, and you can also look for metastasized tumors. You look in the lung, that's where you'd see the metastasized tumor, and look for phospho-SMAD, or SMAD2 in this case. And here we have 4T1 cells in the mice. So these were the original, the tumors in the flank, in the mammary fat pad and in the lung. And if you take these tumors, you grind them up, and you do Western blotting, and you look for phosphorylation SMAD. In this case, the tumor's in the animal. It's being bombarded with TGF-beta from the tumor itself, but the animal's TGF-beta, it's in the bloodstream. So you got lots of phospho-SMAD2. That's what you're trying to block in the first place, right? So there's lots of phospho-SMAD2. You don't serum starve them. You can't serum starve an animal. So there it is, it's phosphorylation. So the tumors are exposed to the serum in the animal's body, lots of phospho-SMAD2. But then the animals that got the interperitoneal injection, that's inside the abdomen, either 10 or 40 milligrams per kilogram body weight, you can actually block the phosphorylation. Here they show two tumors, two primary tumors, 0, 10, or 40 milligrams per kilogram body weight. And then this is the lung nodule, so that's the metastasized tumor. 0 and 40, 0 and 40, these are two tumors. Two untreated tumors and two treated tumors. They block the phospho-SMAD2. So this figure shows that SM16 inhibits TGF-beta signaling in cell culture as well in primary and metastasized tumors in mice. So that was an interperitoneal injection. And that is a very painful way to get a drug into the body, into the bloodstream. So then next, they want to see, can we do this using an oral dose? So in this case, instead of injecting it, they want to see, is it metabolized and does it stay in the bloodstream of the animal long enough for it to take effect? Once again, you implant the tumor in the mammary fat pad. You wait for the tumors to get to 9 to 10 millimeters in area and size. And then you have an oral dose, 0.45 grams, half a gram of SM16 per kilogram of chow. You let the animals eat as much as they want. So it's animal chow that's been infused or perfused with the drug. And then what you do is you take out the plasma to see, is the drug in the bloodstream or not? And two, take out the tumor and take a look and see what happens. And both in terms of phospho-SMAD2, mammary fat pad or metastasized site, which would be the lung. And here are the results. So this is the primary tumors analyzed 24 hours after the chow that's been impregnated with the SM16. The control animals had phospho-SMAD2 because you're not blocking TGF-beta signaling. The SM16, you actually have a nice blockage of the phosphorylation of SMAD2. And then they looked for SM16 concentration in the serum, in the blood of the animals. You see that in the control animals, they didn't have any because they didn't eat any of the chow. But after 24 hours, they had quite a high concentration in micromolar range. Well, after 36 hours and even after 28 days later, they kept eating the chow. They always had this nice constant concentration of the drug in the bloodstream up to a month later. So the figure shows that orally administered SM16 inhibits SMAD phosphorylation on the left in primary tumors and consistently accesses the bloodstream for the duration of the experiment, which was 28 days. So what about tumor size? Oh, what? Did I take that slide out? Sorry, I'm gonna have to escape. Stop share. It's not there anymore. Oh, sorry about that. No punchline there. That's terrible. The point is, if you look at the size of the tumors, they actually shrank. The primary tumors shrank, and the metastasized tumors shrank. I am quite shocked that I did not. Yeah, sorry about that. That is a terrible way to end this talk. But, so basically, yes. Not only does it block the phosphorylation of SMAD2, it actually blocked tumor growth in the mammary fat pad as well as in the metastasized lung tumor. That was great. And that's because it constantly accessed the bloodstream. It was constantly there. And so the second the tumor decided to try to grow, there's this blocking of TGF-beta effects. So, because of that, well, the summary of the lecture today is that serine threonine kinases in cancer. So TGF-beta receptors fall into the following categories: the inhibitors, antibodies, not very promising, because tumors just generate their own TGF-beta, which binds to the receptors where the antibodies can get to them. Kinase inhibitors look very promising, especially that last one, SM16. You have to believe me on the data. And the antisense oligos to ligand are also pretty good, especially in astrocytomas or glioblastoma multiforme. And hopefully I showed you how to characterize a promising TGF-beta receptor inhibitor using multiple different techniques in vitro before you actually go into an animal. And that's my last slide. So question?
When you say that the drug constantly accesses the bloodstream, does that mean like it's not metabolized in the bloodstream quickly or something? Like what does that mean?
So yes, so the drug, so you digest your food containing the drug, it bypasses the liver's ability to, with CYP450 and all these other CYP enzymes, to actually break it down. So it gets into the bloodstream, and then it stays in the bloodstream long enough for it to take effect before your body naturally secretes or excretes it, which it will happen. So when you stop any drug, there will be a time before your body just gets rid of it, right, through excretion. But if you kind of constantly, it's kind of like a chemotherapeutic regimen, right? Every second day you go in with cisplatin, for example, and so that's because cisplatin accesses the bloodstream and stays high enough for 24 hours, and then you go in as it's starting to come down, then you hit it again for the next batch. Same thing with Advil or Tylenol. Take it every 4 hours, every 6 hours, and that's because you get rid of it after about 4 to 6 hours. You're down to 25%, then you can hit it with another batch or another pill, and then it'll come back up. And that's save. Don't save that. I will end this. And it'll be downloaded here. I'll upload. Oh, question.
So for the last slide, you went over the TGF-beta receptor inhibitors in different categories. I'm not sure if you mentioned it last lecture, but what was the reason why there weren't any antibodies generated for the TGF-beta receptor itself?
Oh, no antibodies to TGF-beta receptor. We're trying to get antibodies to the TGF-beta receptor, and we can't make them that work really well. So, in fact, and this is the bane of my students' existence, because we don't have antibodies that recognize type 2 and type 1 receptor that work really, really well. So what we have to do is we have to overexpress receptors that are artificially tagged with flag, HA, V8. I don't know if you've covered any of these artificial tags, MIG tags in class. That's hemagglutinin. And then we go in with an antibody to HA, and that's so that we can actually study the receptor in cells in culture. We try to go with antibodies to type 2 receptor. There are some that sort of work, but it's kind of messy. Type 1 receptor, really hard to find antibodies that work. And yeah, this receptor is really tough to work with, because even antibodies that look for its activity, there are none. So yeah, we're still working with iodinated ligand. So I have to go in with I-125 radioactive ligand. I have to label it in the lab because I don't want my students exposed to it. And we incubate that. So the ligand itself is iodinated. Then we follow the radioactivity of the ligand as it's taken up by cells. And that's the only way we can follow the receptors. Really hard receptor to work with. Great question. Tough receptor, unfortunately. Okay. All right, everyone. So when is the exam again?
24th.
24th. Okay. So you've got lots of time to study. That's good. You have other exams, though?
Yeah.
I hope this is the last class you have. You have more lectures later on this week?
Yeah.
One more? Okay. Well, we'll see you on the 24th because I'll be there. I'll be proctoring with Dr. Danino. And I think what she'll do is she'll probably give you a breakdown of what's on the exam by email or by announcement as well. I don't foresee the exam being much different from what the quizzes are, except like 8 times longer, right? Because it's a 3-hour exam. So it's just a lot more writing involved. You know, design an experiment to test this, those little things like that. Not too taxing while you're trying to, answer questions to try to figure out how to design an experiment, but know your techniques and that sort of thing. So we'll see you on the 24th.